Today, and my job here is to talk about actually one of the pillars of aging, that is proteostasis, and to kind of convey that it goes a little more beyond that cleaning, and I hope I can bring that message. So what is this awful word, proteostasis? So proteostasis refers to the mechanism that maintains our proteins inside our cells healthy. As you know, a protein to be functional needs to be properly folded. And sometimes things happen and the protein unfolds or is not properly formed, and then you have chaperones that are going to take care of refolding the protein and make it again functional. Sometimes this refolding is not possible, and you have to get rid of that protein. And then you have the proteolytic systems to do that. You have the ubiquitin proteasome system that degrades single proteins. And then you have a whole organelle, the lysosomes, that, as we hear from Linda, is able to degrade any kind of macromolecules. This degradation of intracellular components in lysosomes is what is known as autophagy, and it can happen by different mechanisms, depending on how you bring the proteins into the lysosomes. You can just bring them directly across the lysosomal membrane in this process known as chaperone-mediated autophagy. You can just pack them and put them in these multivesicular bodies that will degrade it inside, and this will be called endosomal microautophagy, or they just extrude it outside. And then when they organize in multiple proteins, aggregated, the best way to eliminate it is to create a membrane around, form a vesicle, and then make it fuse with the lysosomes for degradation. And this process is known as macroautophagy. So all these mechanisms, in addition of removing proteins, as I say, goes a little beyond, because basically you can also turn over organelles and pathogens and any other component that is in the cytosol. And then in addition of cleaning, as I will try to illustrate, it has also the capability of relating it with energy production, because you recycle any component that you are degraded in the lysosomes. So what is the relation between proteostasis of aging? Does aging in any way affect proteostasis? So of the multiple experiments from many labs there, I think the most elegant study was done by Rick Morimoto. He works in C. elegans, and he was interested in see how a protein that is called huntingtin, that accumulates in the patients or is mutated in patients with Huntington, aggregates in the worms. So if the protein is not aggregated, you see this very nice soluble diffuse green. And then as you start adding glutamines, that is part of the mutation, you start seeing these very nice aggregates forming. The interesting thing is that all these studies were done with very young C. elegans, with very young worms. But if you get, for example, now this animal that at 4 days of age doesn't have any aggregate, if you just wait, you start seeing how with age, independently that you are not changing the mutation, these proteins start aggregating, suggesting that you are adding an aging component to a protein that is halfway there into going into aggregation, and you just accelerate the process of aggregation, suggesting that aging reduces the ability to maintain protein homeostasis. So if that's the case, why? Why does aging affect proteostasis? So I think that work from many labs now have shown that these proteostasis mechanisms, the chaperones and the proteolytic systems, malfunction with age. And I'm careful in using the word malfunction because it's true that in many conditions you have a reduced function of these systems, but in other circumstances, what you have is a normal, more or less, activity under basal conditions, but then they are unable to do the extra mile to accommodate to conditions in which you have an additional stress. And this is important because these are stress-related pathways. So I think there are examples of failure of these systems for almost every of the components. For example, in chaperones, there is this evidence that you have kind of a chaperone depletion because the increase in metastable proteins with age trap these chaperones in keeping them busy, so you cannot use them for normal physiological processes. In the case of the proteasome, very elegant work showing that there is deregulation in the way that, or the composition of your proteasome, and there has been evidence that this is because the assembly factors that produce this proteasome change with age. In the case of macroautophagy, this process that I mentioned, you trap big chunks of the cytosol or organelles. It's very clear that there is a reduction in the induction of this process, as well as in the trafficking of these vesicles. And this has been linked to alterations in signaling, or more recently to alteration in the motor proteins that have to move these vesicles. And then going into this other form of autophagy that I mentioned, chaperone-mediated autophagy, there is evidence that the transport across the membrane of these proteins to be degraded decrease, and this is due to a problem with the stability of this receptor at the lysosomal membrane. So basically, as I said, there is a lot of research going on in trying to figure out what are the reasons why these mechanisms don't work with age. But what are the consequences? Is this something that we should worry about, the fact that these systems decrease with age? So I'm going to focus for the rest of the talk mostly on autophagy because it's the thing that I know a little better, but almost everything that I'm going to say, you can also find very nice examples for the chaperones and for the ubiquitin proteasome system. This is something common for most of the proteostasis machinery. So autophagy, as we heard very nice from Linda before, has multiple functions in the cell. One of them is clearly quality control, but as I mentioned, it's very important for energetics because you are going to recycle any material, free amino acids, free fatty acids, that you are producing as you break down these components. It has also an important role in cellular defense against pathogens and also for the innate immunity in remodeling anything that requires major changes in a structure or major changes in your proteome, as for example in conditions of reprogramming. So I just mentioned to you that autophagy decreases with age. So thinking on these functions, what you are going to find is a cell that is energetically compromised, that starts accumulating garbage inside, and that is going to be much more vulnerable to insults from the environment and will not have the ability to accommodate to these insults from the environment so easily. So basically, the idea is that autophagy malfunctions in aging, as I mentioned, and this is going to impact, and this is another rendition of the different pillars of aging, it's going to impact proteostasis, because as I mentioned, it's a component of the proteostasis machinery, but also has an impact in other pillars of aging. And this is one of the reasons why I use this model from Felipe Sierra, because I think these connections among the different pillars of aging, and you already hear how much interaction are among the different groups, and as Manolo said, there is no competition, there is cooperation in this case. I think keeping in mind these connections is important. So for example, in the case of autophagy, if you think in addition to proteostasis, as I mentioned, this is a pathway that you activate in response to stress. It contributes to cellular metabolism. It's important for the immune response as a mechanism of defense, and reprogramming is in the basis of cellular regeneration and stem cell biology. So it really impacts in many different aspects of aging. In addition, another strong link in between autophagy and aging has been through these age-related disorders. There is elegant work from many labs showing that autophagy malfunction in many age-related diseases, neurodegenerative diseases, but also metabolic disorders, immune diseases, and the list keeps growing. So then, can we experimentally show that alterations in autophagy are going to affect aging and are going to affect the age-related disorders? So there has been already beautiful examples there, and I'm just going to illustrate something that we are working in the lab. As I said, we are very interested in autophagy, and in particular in this form that is known as chaperone-mediated autophagy, in which the protein, this red protein that you want to degrade, is going to bind to the lysosomal membrane through this receptor that is called LAMP2A, and this is the only protein I'm going to mention in the whole talk. And basically, this forms a translocation complex. The protein goes inside and gets degraded. So a while ago, we found that the levels of this receptor, this LAMP2A, decrease with age. So if you compare in young and old animals, and young and less young people, you can see that there is a reduction in the levels of this receptor protein, and this is something universal. So the idea is that as you get old, your lysosomes are going to have less LAMP2A and will be able to translocate less proteins. So if this is the case, can we just mimic the aging of this autophagy machinery in a young animal and try to see if that affects the aging of the model? So basically, this is what we did, and this is work initiated by Jose in the lab, and then followed by Mathieu. And the idea was let's eliminate or let's mimic unchanges in cell function. So to summarize quickly what I wanted to convey till here, basically malfunction of proteostasis occurs with age. This malfunction or this reduction in proteostasis recapitulates some of the features of aging. If you impose it to a disease, it accelerates the course of disease. And then the positive note, as we also saw from Linda, is that if you preserve autophagy, if you preserve proteostasis, it's going to slow down aging, and also it's going to preserve function, that at the end of the day is what we all want. So in the last two slides, I just want to answer or try to at least throw out there the question of, are we ready to translate this in humans? So, of course, one of the things that we always look at when we want to think about the best way to age is in centenarians. And we have the luxury at Einstein that thanks to the efforts of Nir Barzilai, and you will hear all about that from him, we have this population of centenarians, and that is very nice design because in addition of the centenarians, you have the kids, the offspring of the centenarians, and their spouses that serve as control. So the group of Fernando Macian, in collaboration with Nir and Gil, decide to analyze whether centenarians have beta proteostasis, because they always seem to have everything that is important beta. So of course they measured that in the T cells, in the blood, and they saw that they have preserved proteostasis function and they have great autophagy. And what I found even more interesting is that if they plot the autophagy activity in different individuals against immune function, there is a positive correlation suggesting that this improvement in autophagy nicely correlate with an improvement in immune function. So there is this connection between these two processes. The problem, of course, is we are not all centenarians, and we have to figure out how to do this. So this is kind of my to-do list, or what I think that it's important to start discussing about. So I think regarding of the mechanism of how proteostasis affect aging and the possibility to modulate it and the impact in disease, there has been a lot of advance and progress at the molecular level. We know better the pathways and we know how they are affected. But I think what we are really a little lagging behind is in the integration at the systemic level. So just to illustrate a little what I mean by integration, I think it's very clear to all of us that we are not HeLa cells. And many of the things that we've been doing with single cells probably are not going to have such a big impact when you put it in a whole organism. So most of the studies in proteostasis have focused from years in the individual mechanisms of proteostasis. People study the proteasome, don't talk with people studying autophagy. That fortunately has changed. It's very clear that these systems crosstalk, but also that inside a cell, proteostasis of a mitochondria is coordinated with proteostasis in the nucleus or proteostasis of the endoplasmic reticulum. So we have to think more in this intracellular crosstalk. And of course, if you move a level up, proteostasis in one cell in a tissue is going to be very coordinated with the proteostasis of the neighbor cells because you need an integrated response. And then, of course, the ultimate level, and this has been already shown in model systems, in C. elegans, and in flies, still lacking information in mammals, but there seems to be a clear connection between proteostasis in different organs. And this is in part why we age as an organism and not as a single tissue. So I think we need more and more integration of all this information, cooperative efforts, and of course, money to integrate and to be able to work in all those systems. But regarding mechanisms, I'm quite optimistic that I think these things are going to happen. The other thing that I think we are lacking, or is very important to start thinking, is to understand proteostasis in the general population. I think at this moment we know a lot in animals that are very genetically homogeneous. We have very little information on what is the impact of genetic variability as we have in our population, and whether we can start thinking about personalized proteostasis. Some person might have a particular level that it fits with the rest of her conditions, meanwhile other people might need higher activity. So in order to do that, we really need to develop better methods to measure proteostasis in humans. We are doing very well in animals because you have very invasive methods and you can kill them. We can still not do that in populations, so we really have to develop better methods. And then, of course, And then, of course, the last part will be the interventions. I think you will see through the meeting, and we have a beautiful example with Linda's talk, that many of the interventions that seems to have a positive effect in aging, they have an effect on autophagy and in the proteostasis machinery. So I think once we have these better methods to track autophagy in patients, and many of these interventions are implemented, I think we will start gaining a better understanding on how much this proteostasis is contributing to the good aging or the bad aging in the population. So I think that the next stage is going to be start thinking more in population studies. So with that, I just want to really thank the people in our lab. As I said, a couple of people is here, and it's always great to see them, but I have the luxury to work with a team of extremely motivated people. Our biggest star for this talk is really Imma and her long life animals. We have terrific collaborators in both parts of the Atlantic, and this is the people who gave us the money. Thank you so much for your attention.